Hi, my name is Kirsten, and I'm here to tell you about my dissertation research on horizontal gene transfer in insects. You might have heard the phrase, "you look like your parents." As my mom, that's my dad, and that's me. People always tell me we look alike. This idea that children look like their parents is known as vertical inheritance, or when genes are passed down from parent to offspring. One thing you don't often hear, though, is, "you look just like a jellyfish." But what if jellyfish could actually exchange genes with humans? For example, if they transferred some glow-in-the-dark genes to me, and then I developed that ability. This would be called horizontal inheritance, and it's the topic of my dissertation. Another word for it is horizontal gene transfer, or HGT. People know about HGT in the context of antibiotic resistance because genes pass between bacteria that make infections more difficult to treat. I study HGT from bacteria to animals. My favorite example of this is the tick, which acquired genes that help them suck blood, or the stick insect, which acquired genes that help them digest plants. There are lots of cool examples of this, so I thought I would investigate HGT in my lab model system, a fly known as the wasabi fly, which lives and feeds on plants. After analyzing the genetic sequences of the wasabi fly, we found one that came from a bacteria, a gene called cytolethal descending toxin B, or CDTB for short. This toxin, CDTB, is known to be released by disease-causing bacteria. In this picture, cells on the left are healthy, while those on the right have been intoxicated with CDTB. They are descended and they will eventually die. The takeaway here is that usually CDTB is bad for animals. However, if this is the case, why do we find it in the fly DNA? This paradox is at the heart of my dissertation, understanding how a protein came to benefit the very cells they once evolved to destroy. There is one example of CDTB benefiting animal cells: aphids. Aphids are parasitized by insects called parasitoid wasps, which lay an egg inside the aphid that grows into a baby wasp that eats the aphid alive. However, some aphids have bacteria that secrete CDTB, and it turns out that these aphids that have CDTB are more likely to survive an attack from wasps. So I'm curious whether CDTB, while encoded in the genome of the wasabi fly, or the various other insect species in which I discovered it, is also protecting them from wasps. In order to test this, I'm using gene editing technology to get rid of the gene in flies, and I'm parasitizing them with wasps. If these mutant flies die more often, this will mean that the HGT was beneficial to insects, which could have important implications about the role of HGT in the evolution of insect immunity. I'm still working on these experiments, so please stay tuned to find out what happens. I'd like to thank you for listening and all my collaborators. Thank you so much.